Hello students, welcome to EPG Pathshala. I am Dr. Imtiaz Hassan from Jamia Millia Islamia, Central University. Today we are going to talk on module Fourier transform infrared spectroscopy used in molecular biophysics part 2. After studying this module, the student will be able to understand the basic principle of FTIR spectroscopy. Then we discuss about the components of instruments of the FTIR, and then we discuss about the application of FTIR in different discipline, and at last we discuss about the advanced feature of the FTIR over the dispersive IR spectrometer. Let's start with the basic principle of the Fourier transform IR spectroscopy, that's termed as FTIR spectroscopy. FTIR spectroscopy is a vibrational spectroscopic technique, meaning it takes advantage of asymmetric molecular stretching, vibration, and rotation of chemical bonds as they are exposed to designated wavelength of the light, mainly in the IR region. The Fourier transform is to transform the signal from the time domain to representation in the frequency domain. That's the main advanced feature. And thus FTIR spectrometer obtains an IR spectra by first collecting an interferogram of a sample signal using an interferometer, then performs a Fourier transform on the interferogram to obtain the spectrum. An interferometer is an instrument that uses the technique of superimposing or interfering two or more waves to detect difference between them. So now we can discuss about what is the Fourier transform. You first understand what is the Fourier transform. Fourier transform defines a relationship between the signal in time domain and its representation in the frequency domain. Being a transform, no transformation is created or lost in the process, so the original signal can be recovered from the Fourier transform and vice versa. The Fourier transform of a signal is a continuous complex valued signal capable of presenting real valued or complex valued continuous time signals. In the first row of the graph, the unit of pulse function time and its Fourier transform as a function of W as a function of frequency. Translation, that is the delay in the time domain, goes over to complex phase shift in the frequency domain. In the second row, function G, a delayed unit pulse besides the real imaginary part of the Fourier transform. The Fourier transform decomposes a function into the eigen function of the group of translations. The continuous Fourier transform to use on continuous signal is defined in this relation like as a function of x, and it's given as a 2n into wx, and the inverse continuous Fourier transform, which allows you to go from the spectrum back to the signal and defined in a relation from the integration of function as a function of frequency into 2 pi wtx. So function of frequency is the spectrum where the w represents the frequency and fx is the signal in the time where x represents the time and I is the square of -1. See the complex number theory. A computer can only work with the finite discrete signals, not with the continuous signal. Thus we need to define the discrete Fourier transform. In discrete Fourier transform, the infinite border of the integral can be replaced by a finite ones and the integral symbol can be replaced by sum. So the DFT is defined as function of k equals to 1 by n, and that is the sum of fn e to the power 2 pi kn by n. And the inverse DFT is defined as the sum of the function of k e to the power 2 pi nk divided by n. We discuss about the Fourier transformed IR spectroscopy. The normal spectrum gives you the information about frequency domain, that is the plot of I versus V, intensity as a function of frequency with respect to the frequency, and the time domain, that is the plot of I with respect to time as a function of time with respect to time. So it calls Fourier transformation of the frequency of the spectrum. And each version of a spectrum contains the same information, and conversion of one spectrum from the another spectrum can be accomplished by the computer. So this is showing a typical transmission spectrum of or the Fourier transmission spectrum, which is containing the time domain as well. We discuss transfer interferogram to absorption spectrum. You can see here time axis by fast Fourier transformation converted into the wave number, and it is done by Michelson interferometer, which is based on the interface of the waves. Like you can see here, if the wave are in the same phase, it gave result in the deep wave. It is the constructive interference. However, wave are partially out of phase, it results in a slight destructive. But if waves are in opposite orientation, completely out of the phase, it leads to the destructive interference and the result is zero. Now we talk about the Michelson interferometer, a simplest interferometer design, and which having beam splitter for dividing the incoming IR beam into the two parts. The two plane mirror for the reflecting the two beams back to the beam splitter, where they interfere either constructively or destructively depending on the position of the moving mirror. The position of moving mirror is expressed as optical path difference, and the optical path difference is the distance traveled by the red beam minus distance traveled by the yellow beam, as given here. Now it talks about the component of FTIR, it having simple light source, that IR energy is emitted from a glowing black body source which ends with the detector, and it is coupled with the interferometer, that's beam enters to the interferometer where the spectral encoding takes place by, and the interferogram which signal then exits the interferometer, and the beam splitter which takes the incoming beam and divides it into two optical beams. Then the sample beam enters to the sample compartment where it is transmitted through or reflected off the surface of the sample. Then detector, the beam finally passes to the detector for a final measurement, and of course you need a computer which measures the signal and digitized and sent to the computer where the Fourier transformation takes place. And moving and fixed mirror in the interferometer is the only moving part of the instrument. We discuss how FTIR spectrometry is created. The interferogram contains information over the entire IR region to which the detector is responsive. A mathematical operation known as Fourier transformation converts the interferogram, a time domain spectrum displaying intensity versus time domain of the mirror scan, to the real IR spectrum, which is the familiar frequency domain spectrum showing intensity versus frequency. The detector signal is sampled to small precise intervals during the mirror scan. Control of the sampling. The sampling rate is controlled by the internal independent reference, a modulated monochromatic beam of a helium. A neon, hydrogen neon laser focused to the separate detector. You can see here the time domain intensity versus OPD per centimeter. After Fourier transformation pair, it gives you a sharp intense peak by the Fourier transformation in the form of intensity versus the wave number in the frequency domain. We discuss about the Fourier transform IR spectroscopy, the relationship between the light source spectrum and the signal output from the interferometer. The light source is the monochromatic light, dichroic light and continuous spectrum light are of different type, and it's giving the different types of output from the interference wave after the Fourier transformation. Now we talk about the FTIR detectors. The two most popular detectors for FTIR spectrometer are: one is the deuterated triglycine sulfate, named as DTGS. It is a pyroelectric detector that delivers rapid response because it measures the change in temperature rather than the value of temperature. It operates at room temperature. The second is the mercury cadmium telluride, MCT, is a photon or quantum detector that depends on the quantum nature of the radiation and also exhibits very fast response. It must be maintained at liquid nitrogen temperature around 77 Kelvin to be effective. In general, the MCT detector is faster and more sensitive than DTGS detector. There are some thermal detectors which are all detectors are not used in the FTIR. The response time of the thermal detector, for example, thermocouple and thermistor used in dispersive IR instruments, are too slow for the rapid scan time, 1 second or less of the interferometer. We discuss the sequence of obtaining IR spectrum. You can see here in the figure that interferogram with the nitrogen, background peak and the transmission spectrum. Similarly, interferogram with the sample, single beam spectrum and absorbance of the spectrum. So the transmission spectrum is a single beam sample divided by the background. Absorbance spectrum equals to the negative logarithm of the transmittance and automatically converts and displays spectra as a absorbance spectra. The interferogram of background is obtained without the sample. System uses Fourier transform to create single beam background spectrum. Interferogram of sample is obtained and system uses Fourier transform to create single beam spectrum of the sample. And finally, system calculates the transmittance or absorption spectrum in the form of FTIR, which having a useful information about the molecule. We discuss about the relationship between the optical frequency and interferometer frequency. OPD equals to 2 into mm minus fm equals to sigma retardation. When output power from the detector plot versus sigma, it gives the interferogram. Frequency of the interferometer resulted less than the source frequency. So relationship between the two frequencies is given as 1 cycle of signal occurs when the mirror moves at a distance of lambda by 2, and assuming the constant velocity of mm, of vm and tau as a time required for mirror to move pi by 2. So Vm tau equals to pi by 2. The frequency f of signal at a detector is the reciprocal of tau, where frequency f equals to 1 by tau equals to Vm by lambda by 2 and equals to 2 into Vm by lambda, which is further derived as f equals to 2 Vm nu and lambda equals to c by nu. So finally f equals to 2 Vm by c into v, where v is the frequency of radiation and c is the velocity of light. At constant Vm, f interferogram frequency is depend on the optical frequency nu. Now we discuss about some terminology that are mostly used in the FTIR, like the resolution. That's the common definition is the separation of various spectral wavelengths, usually defined in terms of wave number. So it gives you the idea that how the peaks are separated by each other. The setting of 4 to 8 per centimeter is sufficient for most solid and liquid sample. In the gas analysis experiment, may need a resolution of 2 to 2 centimeter or higher. Higher the resolution, experiment will have lower signal to noise ratio. The resolution, the some FTIR scanning parameters, two widely spaced line like the taking data or a short path difference is sufficient to resolve these lines. The two close lines, the interferogram must be measured over the longer path difference to get the satisfactory spectrum. The two closely spaced line only separated if one complete beat the record, as lines get closer together and delta must get increases. It is given as delta nu per centimeter equals to 1 by sigma, where the mirror motion is 1 by sigma and resolution is governed by the distance as the travels. We can see here the advantages of FTIR over the dispersive IR, and we compare one by one that for the dispersive IR, many moving parts results in a mechanical slippage. However, in the FTIR, only mirror moves during the experiment. In dispersive FIR, calibration against reference spectra is required to measure the frequency. But in FTIR, use of laser provides high frequency precision in the order of 0.01 per centimeter. That's the cone's advantage. In the dispersive IR, stray light within the instrument causes spurious reading. However, in the case of FTIR, a straight light does not affect detector since all signals are modulated. In dispersive FIR, in order to improve the resolution, only a small amount of IR beam is allowed to pass through the slits, but the FTIR have an advantage that much larger beam aperture used and high frequency throughout the jacket's advantage. Only narrow frequency radiation falls on the detector at any time in dispersive IR. However, all frequency of radiation falls on detector simultaneously, and improved signal to noise ratio obtained quickly. That's the multiply or Falaget's advantage in the case of FTIR. In dispersive IR, slow scan speeds makes dispersive instrument too slow for monitoring system undergoing rapid change. But in FTIR, rapid scan speed permits monitoring sample undergoing rapid change. In the dispersive IR, sample subject to thermal effects from the source due to length of scan time. In the FTIR, short scan times, hence sample is not subject to thermal effects. In dispersive IR, any emission of IR radiation by sample will falls on detector due to the conventional positioning of the sample before the monochromator. But in FTIR, any emission of IR radiation by sample will not be detected. And finally, in the dispersive IR, double beam optics permits continuous real-time background subtraction, but in FTIR, single beam optics background spectrum collected separately in time from sample spectrum can result in error if background spectra are not collected. So now you are able to know what are the advantages of FTIR over the dispersive IR. A comparison between dispersion spectrometer and FTIR spectrometer is shown here. You can see in the figure that the separate IR light and grafting is used in the dispersive IR. In order to measure the IR spectrum, the dispersion spectrometer takes several minutes. Also, the detector receives only a few % of energy of the original light source. However, in the case of FTIR, in order to measure the IR spectrum, FTIR takes only few seconds. Moreover, the detector receives up to 50% of the energy of original light source, much larger than the dispersive spectrometer. And the infantogram is calculated and transformed into a spectrum using the Fourier transform, and infantogram is the first made by the infantometer. Let's talk about the advantages of FTIR. The first advantage is that the cell gets multiplex. That is the advantage having high signal to noise ratio as compared to the dispersive instrument. The FTIR collects all resolution of element with a complete scan of interferometer, and the successive scan of FTIR instrument were coded and averaged to enhance the signal to noise ratio of the spectrum. As the number of scans increases, the signal to noise ratio is increases, and data quality of course enhance very well. Theoretically, an infinitely long scan would average out all the noise of the baseline. However, it is not possible, but it can be theoretically done. That's what we focus to increase the number of scans as much as we can. The dispersive instrument collects data one wavelength at a time and collect only a single spectrum, and there is no good method for increasing the signal to noise ratio by the dispersive spectrum. However, we have a very FTIR coupled system interferometer which gives us a very high data quality with an improved signal to noise ratio. The second advantage of FTIR is the cons advantage. That's the high resolution, reproducibility, and highly accurate frequency determination. This technique allows high speed sampling with the aid of high laser light interference fringes. It requires no wave number correction and provide wave number to an accuracy of 0.01 per centimeter. The third advantage of FTIR is much higher E output, that's known as Jackie note or throughput advantage, because not using classical monochromator. It requires no slit device, making good use of the available beam. The fourth advantage is the better sensitivity. In the interferometer, the radiation power transmitted onto the detector is very high, which results in high sensitivity. That allows simultaneous measurement over the entire wavelength range. The fifth advantage, no stray light in the FTIR. FTIR allows only interference signal to contribute to the spectrum. Background light effect greatly lower, and it allows selective handling of signal limiting interference. The sixth advantage is the wave number range flexibility and simple to alter the instrument wave number range in FTIR. Apart from all these advantages, FTIR should also have some disadvantage, like the single beam. It requires collecting a blank. It cannot use thermal detector because in thermal detection is too slow process. CO2 and water are highly sensitive. It is destructive, causes destruction, and too sensitive that it would detect the small contaminants. That's what these are some disadvantages associated with the IR. It also has a lot of limitation. For example, with IR spectroscopy, it is not possible to know the molecular weight of substances. Although it's everything is dependent on the mass, but exact molecular weight is not calculated by the FTIR, and it is frequently non-adherence to the Beer-Lambert law of complexity of the spectra, and the narrowness of the spectra and effect of many stray radiation make the measurement of absorbance upon slit width and wavelength settling. Now we talk about the application of FTIR. FTIR is widely used in different areas, like in chemistry it is used for the identification of functional group. It is also used for trace analysis of raw materials of finished product. For example, if you developed a product and you want to check it should not have any contamination, then you scan for that particular wavelength. If the peak is not appearing for that contaminant, it means the sample is fresh. Or you can compare with the known IR spectrum of the fresh sample with the other sample. It is also used for the analysis of solid, liquid and gases, kinetics of reaction on a microsecond time scale, like the appearance of product, appearance of intermediate and disappearance of reactant at a different time scale. It is also used for the analysis of chromatographic and thermogravimetric sample fractions. Then the substance of weak absorption sample for the IR emission ready studies. It is also used in the forensic lab. A forensic scientist would use the FTIR to identify chemicals in the different sample like the paints, polymers, coating, rug, contaminant and explosive residues. We discussed the uses of FTIR in soil analysis. You know that the FTIR is having a large number of applications in all field of science. Here we discuss about the use in soil analysis. You can see here the three soils like the calcium carbonate, bentonite and kaolin. The IR spectra of these three types of soil are quite different. The kaolin having a strong peak at the 1000 wave number, but bentonite having a very low peak at the 1000 wave number. But the calcium carbonate soil have an intense peak at the 1500 wave number. So FTIR is used to estimate the different quality of the soil. This is the another spectra showing the IR spectrum of the different types of soil, like the gromosol shown in the magenta, hamra soil in the green, terra soil in the red, loess soil in the cyan, mesum soil in the violet, red soil in the light green, rice soil and different types of soil. So by IR spectra you may be able to identify and quantify and you check the quality of soil as well. Here we discuss the another important use of FTIR in the fuel analysis. You can see the spectrum is recorded as a mild in the mid IR region using FTIR spectrometer. About 40 parameters are recorded in a single measurement within the few seconds, and you can see here the FTIR spectra of aromatics, MTBE, olefins and oxygenates are quite different, so you can analyze the fuel quality by using IR spectrometry. Not only this, you can also analyze the combustion product analysis. Various absorption band of different chemical groups of combustion products are seen in FTIR. You can see here that the combustion product of methane are found give a peak around 3000 wave number, but combustion peak of carbon monoxide and carbon dioxide is around 2000, and again the carbon dioxide having a very low frequency. So by this you can identify the combustion product as well by using FTIR. We discuss use of FTIR in biology. Apart from its use in chemistry, physics, material science, FTIR is widely used in the biology as well. It is widely used for the tissue imaging of different spatial resolution. FTIR is also coupled with the different types of microscope to do the microscopic analysis. Here you can see the infrared milk analyzer to determine the fat, protein and lactose content in the milk, measurement of degree of unsaturation of fat and oils, identification of flavor and aroma. FTIR in combination with GC can be used to analyze and evaluate the essential component from the cinnamon bark. For packaging film to identify structural changes occurred due to the exfoliation and interaction of layered silicate in the polymer matrix. You can see here the IR spectra of different types of food product like the protein and lipid only, protein only, oleic nucleic acids and carbohydrates. And at last FTIR average spectra of the TCY70 and TCY in the dotted line is giving from the different strain of the yeast. We discussed the characterization of protein delivery system in real-time PCR. Using the fiber optic based probe, you can take the polymer and you can check the IR spectra of the protein. Real time exponentially increases release of the model protein. As the, for example, here the human serum albumin was observed from the two different polymeric hydrogel formatted with the pH sensitive cross linker. So the pH sensitive cross linker carrying protein, but once the protein released inside the cell in a time dependent manner, the peak of protein intensity is increasing, and that's what you can take the advantage of real time FTIR to see the amount of protein released from the hydrogels in a time dependent manner. It gives you the idea about the structural information on protein can be derived from the FTIR. Now it is most commonly technique for the getting the structural information of the protein, and you can see here the CO bond gives you the amide 1 vibration bands, and NH group gives the amide 2 vibration, and here you can see the amide 1 and amide 2 peaks, FTIR peaks. So FTIR spectrum of bovine pancreatic trypsin is showing amide 1 and amide 2 bands, and B is showing results of Fourier self-deconvolution analysis of amide 1 band of bovine pancreatic trypsin inhibitor. The assignment of various peaks to the different elements of the secondary structure are shown. In the right panel you see an intense band of the amide 1 bond and amide 2 bond for deuterated BPT, bovine pancreatic trypsin inhibitor. The course of hydrogen deuterium from BPT1 from 3 minutes to 374 minutes after initiating the exchange. Amide 2 band at 1450 per centimeter is for deuterated BPTI and is increasing with the time. The amide 2 band at 50 per centimeter is for hydrogenated BPTI and decreasing with the time. The bottom red spectrum at 1550 per centimeter was obtained after complete exchange. The residual absorbency in this region is due to the group of protein other than amide group. So FTIR is extensively used to know the structural content of the protein. Not only this, the change in the structure, change in the conformation, and change in the dynamics of the protein can also be measured by the use of FTIR. We discuss about the structural information on DNA. The FTIR can also be used to get the structural information of DNA. You can see here that the bDNA is converted into the zDNA, and you can see here the FTIR spectra of the zDNA and bDNA are quite different. So the FTIR spectroscopy for monitoring the DNA signal in cell as a histone acetylation was regulated by trichostatin TSA, demonstrating that a significant transformation of bDNA to zDNA upon histone acetylation in the HeLa cells. So FTIR is not only used to see the structural information on the particular type of DNA, but it also used to see the dynamics or the conversion of one form of DNA to other form of DNA upon the chemical modification, and that can easily be identified by FTIR spectroscopy. It is also used to identify the different types of diseases. You can see here the normal person's IR spectra are shown here, but if it having the migraine, the IR spectra are different. In the Duchenne muscular dystrophic patient, in the paralytic patient, in the epileptic patient, and in the Alzheimer's disease, the IR spectra are quite different, and that what the IR spectroscopy may also be used for the detection of diseases. So students, let's now summarize what we have learned in this module. First, the absorption peak in an IR absorption spectrum arise from the molecular vibration of the atom, either it is because of the stretching or because of the bending. Then the Fourier transform is to transform the signal from the time domain to its representation in the frequency domain. That's the advantage of the FTIR. And the heart of FTIR is the Michelson interferometer. The interferometer, which modulates radiation emitted by an IR source, producing an interferogram that has all IR frequencies encoded into it. The interferometer performs an optical Fourier transform on IR radiation emitted by the IR source. The Fourier transform of a signal is continuous, complex valued signal capable of representing real valued of complex valued continuous time signals. The whole IR spectrum is measured at a high speed by the FTIR, and a spectral range is continuously calibrated with the helium and neon laser, and FTIR is a very fast, extremely accurate, and provides very accurate measurements, and it is widely used in large number of different discipline like the chemistry, physics, material science, nanotechnology, clinical science, and biological science to get detailed information about the molecular architecture and the changes in the environment or impact of environment on the molecules. So with this, I finalize this FTIR module. Thank you.